Hi, this is Mary from Open Helix with this week's Tip of the Week. Today I'm going to be talking about a tool called Melina II that's provided by the University of Tokyo Human Genome Center, but I won't be able to cover everything that's possible to know about Melina II in this short movie, so I encourage you to check out our blog at blog.openhelix.com for more details about the Melina II tool that I'll be describing, some references associated with that, and some additional details. We also have tips of the week every week, so be sure to come and check us out. But let's move right now over to the Melina II tool to get started. So I wanted to present Melina II today because at a workshop last week I was describing this tool to someone who had a need for promoter analysis. And this is a very nice web-based tool for promoter analysis. What I'm going to do is illustrate how to use Melina II with the sample set of sequences they provide. This is their basic interface. I'm going to click the sample here and load up this 200NR query sequence set. So here's some sequences that I suspect might have some motif in common, or maybe I'm hoping they have some motif in common. I can enter the sequences here. I can then choose a number of different algorithms to analyze these sequences and see if there are some promoter motifs available. And there are 5 different tools you could actually choose. There are 4 shown by default, but you could come in and adjust them all. Another thing that you could do is you could run the same exact algorithm with a little bit of change in the parameter by just going in and setting the parameters here. For example, if I wanted to run consensus and change the parameters here, I could do that. I'm going to leave the default settings though. And you could change the next one to consensus and change those settings a little bit and do that. For our purposes, I'm just going to leave all the default settings here. I'm going to run these sequences and click Submit. What you'll see is you get a job ID that you could use later to come back and retrieve your results and look at them again at some other point. But right now, we're just going to wait for the results to appear, and here they are. So here are the sequences that I entered. And here is about a 200 base pair region upstream of my starting with methionine, let's say here. And here you can see that I have discovered motifs with these different tools. Now I can click on any one of these motifs, and what I see is that the Gibbs motif here has found that this sequence is a motif that's been discovered in more than one of the sequences. I can look down at the motif sequence set here, and I can look across the different sequences that were input and look at those sequences and look at the adjacent sequences as well. I can look at this motif with the web logo format that's illustrated here, or you can access this as a weight matrix as well, which might be helpful if you wanted to take this matrix to other tools and explore other tools with this kind of defined matrix. But I'll just leave it back set at the sequence logo format right now. The next thing you can do is take this information from here and perform some more analysis on them. Here you could choose the matrix DB or the JASPAR data set, or you could choose genome DB here and choose to run this sequence in the context of several different species here and see if there were other promoters with this motif identified in those data sets as well. So I think this is a really nice graphical interface that describes promoters that may have been found with these different algorithms all in one place. Now you could run each of these tools individually at their home locations, but I just think this is a really nice way to get started, especially if you don't know exactly what you're looking for. You want to enter some sequences, see if there's anything interesting that comes up, and then you can start analyzing them in more detail. You could start changing the parameters at any time, and there's plenty of other things that you can do once you've started to look around a little bit. Let me just give full and total credit to the folks who developed Melina here. This is the Human Genome Center at the University of Tokyo, and you can have access to the individual resources that were used, and also you'll have references that are associated with Melina II that you can find on their help page. So let me just bring you back to the Melina II page and encourage you to come over and try this out if you're looking for motifs that might be present in your DNA sequences. Thanks very much for your time.